All right, good morning. So today we're going to finish the renal system. Last week we finished talking about the last portion of the nephron. We said that the DCT was our last chance to pull back sodium. Now we're going to look at where all our nephrons are actually dumping into, which is the collecting duct. And now we're going to be moving back down the medulla again, so deeper into that corticomedullary gradient. And so we're going to be picking up reabsorbing water into the vasa recta. We're going to do that through aquaporins again, just like we did in the descending limb. But these are going to be aquaporin 2 instead of 1, so make sure you have those differences down. And then this is the last chance we have for any reabsorption at all. So this is the last chance for the water. After this, it gets dumped into a renal calyx, and then to the ureters, to the bladder. We do no more absorption. So everything after this is just urine. It's what you're going to excrete. Now that we're done talking about our reabsorption system, we could talk about modulating it, specifically how we can help maintain proper blood pressure. And we'll talk about it in terms of some drastic, somebody got stabbed, it's an emergency story, but I want you to recognize that this is just a normal system we use all the time. This is going to be called the renin-angiotensin-aldosterone system, or the ROS system. It starts with these juxtaglomerular cells in the afferent arterioles. So these are going to be in the endothelial cells, not the smooth muscle around it like we talked about before. And this is going to be monitoring blood pressure. In our vessels, the blood pressure is the amount of pressure on the wall. So this is also a function of volume. Okay, so if you have a smaller radius or higher volume, you have higher blood pressure. If you have low blood pressure, which you can also think of in this case as low volume, you are going to release renin. Renin is coming out of these juxtaglomerular cells and acting as a proteolytic enzyme in the blood. So that's going to cleave off a portion of angiotensinogen. This is a plasma protein that we always have, and it's inactive unless we clip off portions of it to make it angiotensin 1. Now, angiotensin 1 probably has a function. I can't imagine why we use it as an intermediary if it does, but no one seems to know what it does. So it's not really important to our story. We're just going to say angiotensin 2 is then going to get converted to angiotensin 1 will get converted to angiotensin 2 via ACE, or angiotensin converting enzyme. So we talked about this before as well, but we were talking about ACE2 in the lungs that could bind the COVID virus, the SARS-CoV-2. This is ACE1. I'll draw that in there. We didn't used to have to specify the difference because we never used to talk about ACE until coronavirus. But now this is ACE1 converting that angiotensin 1 into angiotensin 2, and this is the important player. Okay, this is what's really going to make a difference in the system. Angiotensin 2 can both cause vasoconstriction, so binding to smooth muscle around arterioles, arteries, venules, and veins, and decrease the radius, which is going to increase that blood pressure. And it can also cause aldosterone release. Okay, so this is going to lead us into sort of a second portion of the story. The aldosterone is a steroid hormone in the adrenal cortex. Like all steroid hormones, this is going to be able to bind to cytoplasmic receptors because it can pass right through a membrane. It's going to find those receptors in the distal convoluted tubule, and when it binds to them, it's going to act as a transcription factor, and it's going to upregulate the amount of epithelial sodium channels and sodium-potassium ATPase channels that we have in the DCT. So those were our apical and our basilar channels that we use to push sodium into the cell and then back out so that we could get it into the peritubular capillaries. This is going to increase the amount of sodium you have in your blood, meaning it's going to increase blood osmolarity. Okay, that blood osmolarity can then be a signal for this third portion, because the hypothalamus has osmolarity sensors. If it senses high osmolarity, it's going to react by causing the posterior pituitary to release vasopressin. This vasopressin, it's an endocrine hormone. It's going to go through your blood system. Eventually, it's going to get to the nephron, and it's going to find receptors on the collecting duct. So those GPCRs can bind to vasopressin, and it's going to cause an increase in exocytosis of these aquaporin channels. So you're going to put more aquaporin channels into your collecting duct epithelium. When you do that, you're going to move more water across, so you're going to increase your blood volume. Increasing the blood volume will help increase your pressure. Okay. So let's just start by finishing up with the collecting duct. And its reabsorption. So the collecting duct is that structure that's going to have 5 or 6 different nephrons that dump into it. So if we draw out our favorite little nephron, this last portion is our collecting duct. So if I draw this out here, it's going to look just like every part of the nephron that we drew. It's going to have a single layer of this epithelium. We can orient it. We'll have a basilar side sitting on a basal lamina. We'll have vasculature that's going to surround it. But because this is moving down the medulla, this is going to be vasa recta. Okay, so you would have filtrate coming from the DCT. It's going to come around the corner and start moving down the collecting duct. So when we're at this cortex region, the osmolarity out here in the interstitial space is going to be about 300 milliosmoles. And as we move down the medulla, we're going to increase to about 1200 at the very bottom. So way down here, we're closer to 1200 and about 300 up here. If we're moving filtrate down deeper, that means it wants to come to equilibrium with a much higher concentration. We're trying to concentrate urine here. And we do that by allowing water to be reabsorbed. If we pull that out, then we have more solute, less solution. We've increased the osmolarity. So we are going to use green. We'll use green. So we've got to have receptors to move that. And we've got to have one on the apical side, basilar, to move it into the cell, and then one on the basilar side to move it out into the interstitium. And it's going to be the same on both sides, aquaporin 2. So whatever water you have left in the system, you're going to push it into the cell, push it back out, pick it up in the vasa recta, and then the vasa recta is going to be moving counter current in the opposite direction so that it can take that water away and maintain the gradients. If this works well, you're going to actually bring your urine concentration back down to 1200 again, having pulled out pretty much everything you can, and then that is going to leave. So you'll have pretty low volume if you concentrate because you've pulled most of it back. If you have dilute urine, you have a higher volume. Okay. So dilute urine can either mean you're very well hydrated, so you've already pulled everything you can out and there's still excess water. So you're going to pee more and it'll be dilute. Concentrated urine is usually dehydration, or you're just doing a really good job pulling back what you needed. Okay. So questions on the collecting duct. I want you to remember this difference, aquaporin 2, because when we talk about the descending limb aquaporin 1, those are static. We don't change them much. They'll wear out eventually and get replaced, but we're not changing the amounts constantly. Here in the collecting duct, we can change the amount of these aquaporins. We can do something we call upregulation, which means we're plugging more of them in. We put more channels into the membrane. We can also downregulate, meaning we're going to endocytose them and destroy them. Okay, so we'll talk about the modulation of that. But by doing that, we can control how much water we're reabsorbing versus how much we're sending out in our urine. Okay. So for the renin, ouch, angiotensin-aldosterone story, we're going to start again by looking at a big picture of the nephron. But then I'm going to kind of close in on it for each portion. And I will try to keep this from getting too messy. But it's going to be hard because we're going to be talking about a lot of different systems. All right, so here's our nephron. Bowman's capsule, proximal convoluted tubule, descending limb, all very important, but we're not including them in this story, so ascending, distal convoluted tubule, and I'm going to draw it the same color. This is collecting duct. So we can label some things, DCT, PCT. I'll label that as BC for Bowman's capsule, CD for collecting duct, and we'll say descending and ascending limbs. But we also have our vasculature that's working in conjunction. So we've got an afferent arteriole that comes in to the glomerular capillary to an efferent arteriole. So this is efferent. And up here is going to be afferent. In between, of course, this is your glomerular capillary. But in the afferent arteriole, within the endothelial cells that are lining this, we're going to have a certain amount of cells that are called juxtaglomerular cells. Juxtaglomerular. Okay. So your juxtaglomerular cells are your baroreceptors. Glomerular. These are the ones that are going to monitor stretch on the walls of the arterioles. We didn't talk about baroreceptors. We've got them throughout the body. We use them for a number of things. And essentially what they do is they're a pressure sensor. Okay, so they'll be triggered either by pressure on it or by release of pressure on it. In this case, it's release of pressure. Normally, you would expect to have enough pressure on the walls from that hydrostatic pressure to be able to push fluid from the glomerular capillary to Bowman's. If you don't have that much pressure, then you're not going to trigger these juxtaglomerular cells. So, a decrease in blood pressure, which you can also read as volume, because the volume of the blood is what's going to help you put more pressure on that wall, causes the release of renin. So we're going to have molecules of renin that get released from these juxtaglomerular cells directly into the blood. Renin is a proteolytic enzyme. So proteo means protein, lytic means to lyse, to break. So this is going to be an enzyme that's specifically breaking or cleaving another protein. What it's going to affect is something called, let's do blue, angiotensinogen. I don't want to write it there. Oh, screw it. We'll just write it this way. Angiotensinogen. That O-gin on the end is telling you that this is inactive. It's an inactive form. We'll talk about these in digestion too. A lot of times you have proteins that have an effect, breaking things down. You don't want them just willy-nilly going around doing that. You want to control it. So you set them out as an inactive form and only activate it under certain conditions. So angiotensinogen is a plasma protein. Meaning it's just a protein that hangs out in the blood all the time. It could do that safely because it's inactive unless it binds renin. So when you've got this renin that gets released into your blood, the renin can bind to angiotensinogen and it will cleave a portion of that angiotensinogen off and create angiotensin 1. Oh, that's terrible. Okay, so I think it makes more sense. I like to draw it out this way. You have angiotensinogen as your start. You're going to convert that into angiotensin 1 using renin. So that's your enzyme that does this conversion. Angiotensin 1 has no role in this story beyond just being an intermediary. Okay, so we're just going to say from that, we're now going to convert it again. So we'll cut off a portion of the angiotensin 1, and that's going to make angiotensin 2. What we're using for angiotensin 2 is ACE. Specifically ACE1. So I'm just going to put angiotensin 1. This is an intermediary. ACE, or angiotensin converting enzyme, is mainly from the lungs, just like ACE2. So ACE2 we said was that membrane receptor that we found on lung tissue, and the coronavirus could bind to it. So COVID could bind, and we weren't entirely sure what happened after that, but that was how you got infected. ACE1 is an isomer, but it actually acts in almost the exact opposite way. So this is not going to have anything to do with the ACE2 story. This cannot be bound by COVID. And when we talk about ACE actually breaking down angiotensin into angiotensin 2, ACE2 can't do that. It does the exact opposite. So make sure you're clear on ACE1 and ACE2. Once it converts to angiotensin 2, that's the big one. So now we've got angiotensin 2 in our blood. And that's going to go through your entire system and bind anything it can get to. This is going to act as a vasoconstrictor. So this binds to smooth muscle around vasculature. told that story before. We said we had these macula densa cells in the DCT that released adenosine and bound to smooth muscle around the afferent arteriole. We decreased that radius, we decreased flow rate. That was the story on Friday. This is different. This is not being released as a paracrine to just diffuse locally. This is endocrine. It is in the blood. All of this happened in the blood. And now we've got angiotensin II running around our entire body. Any receptor it can find on any smooth muscle around any blood vessel, it can bind and causes vasoconstriction. So we're causing global effects with this. And this is pretty potent too. But you can imagine if you are closing down the radius of your blood vessels throughout your entire body, that you are going to be reducing the volume of the container that holds the blood. Okay? When you do that, you'll have more pressure, because even with the same amount of blood in the system, you're going to have more of it in contact with the wall, putting pressure on it. So if we told this story, not just as this is an everyday modulation that we can use to kind of increase and decrease the blood pressure, but in an emergency situation. We're going to go back to that poor guy that got stabbed in the chest and it collapsed his lung because it opened up his pleural cavity. But that's not the only thing that happened, because when you stab someone like that, you're guaranteed to cut a few arteries and capillaries and everything else. So he's bleeding too. Bad. Like they may have cut something big. And as he's bleeding, he's losing blood volume. Outside, inside, whatever, it doesn't matter. If it's not in the container anymore, you've got a problem. Eventually, you'll run out of it if no one stops it. But right now, the biggest problem is that as you decrease that volume and decrease the pressure, you don't have enough pressure to move the blood through your system. So what blood you have left is not even moving around and supplying oxygen to the areas it needs to. The first thing you need to do is whatever you can to get that blood pressure up, and so you still have what blood you have left moving around. That's this story, angiotensin II. This is short term, just going to collapse all of those arterioles and arteries, making that radius smaller and increasing the blood pressure. But this is not sustainable in the long term. You can't, like if you're actually losing blood pressure like that, you can't close these down all the way all the time. So this is just enough to keep you going until we get to the next portions of the story. Because the second thing that angiotensin II can do is it can bind to the adrenal cortex to cause release of aldosterone. Maybe we can fit it in here. Let's try to do it here. So we said that the adrenal gland was that little thing that was on top of your kidneys. So if I just drew a kidney, and this is actually properly oriented, this is how it would look in the body, unlike how I normally draw it, you would have these adrenal glands sitting on top. I'm going to draw the adrenal gland bigger here. And the adrenal gland, just like the kidney and just like lots of our organs, actually has two portions. There's the cortex, which is the bark that goes around on the outside. And then there's the medulla, which is deeper inside. Today, we're talking about the cortex. We release different things from different portions, but aldosterone comes from the cortex. So you'll have vesicles that have all of this aldosterone ready and made to go. And you're going to have receptors. So now you've got angiotensin II. It's running through your blood. It's causing all this vasoconstriction, but it's also coming to the adrenal gland and binding to the adrenal cortex, and it's going to cause the exocytosis of those vesicles, which releases the aldosterone. Oops. Aldosterone will then get picked up into the blood, and it's going to travel through your body. A reminder that aldosterone is a steroid hormone, and when we talk about steroid hormones, we always know that they're going to be hydrophobic and that they can bind to cytoplasmic receptors. In this case, aldosterone's receptors, at least for the story we're telling, this is going to be in the DCT. So it's binding at the DCT. Which means it goes through this blood, it goes through the entire system. Eventually, it's going to get to the afferent arterial. It's going to pass through the glomerular capillary, efferent arterial. This has a branch off of it as the peritubular capillary that winds around your convoluted tubules. Okay, so you will eventually get this aldosterone over here to your DCT. So let's draw our DCT again. That's a DCT epithelium. And we'll say this is the basilar side. So that's facing, I'm going to have to erase my kidney. The peritubular capillary. So as your aldosterone is passing through the blood, it can get out of capillaries and then pass right through membranes. So this aldosterone is going to go directly into the DCT cell. And now you've got aldosterone. This is going to be binding to that cytoplasmic receptor. We always draw it like it's some kind of box. Of course it's not. It has a 3D structure. But it has a hormone binding domain. And once that's activated, then it has a nuclear access domain that lets it get into the nucleus. Has a DNA binding domain that lets it actually bind to the DNA. And then the transactivation domain, which allows transcription factors to bind to it so that it can influence that transcription. So we'll say here's our nucleus, and that's really not to scale. So we're just gonna there's our cytoplasmic receptor with our hormone bound to it. And of course, we have our DNA in here. So you bind to this and you increase the transcription of certain genes. Aldosterone is going to increase transcription of the sodium channels. So in the DCT, we had sodium channels on the apical side that were epithelial sodium channels, or ENaCs. And then on the basilar side, we had sodium-potassium ATPases. So this aldosterone puts more sodium channels into your DCT. Now you've got a whole bunch more receptors here on both sides of your cell, which I didn't even draw. Oops. But there you go. You could draw it here too and just say, yay, we got all of these receptors now. So if you have more receptors, it's easier to pick up more sodium. The sodium that was in the filtrate can now move through, pass through the channels, and then get picked up into the paratubular capillary. So the function of this aldosterone is to increase sodium reabsorption. Now, anytime you put more solute into a solution, you've increased the osmolarity. There's more solute to create a pull. In this case, you're really going to do it because if we're still telling the story about this poor guy who's bleeding out, he's losing all of his solution and we're sticking more salt in there. Doesn't really do him a whole lot of good short term. But the fact that this is increasing the osmolarity will help. Because now we can sense that high osmolarity in the brain. Okay, so there was aldosterone. It created higher levels of osmolarity. And now we've got our brain. And we have a hypothalamus. And that hypothalamus is in communication with the pituitary. But we said that the pituitary has two separate regions. One is the anterior pituitary and one is the posterior. So the anterior is the one we really focused on when we talked about endocrine. We said that hypothalamus would releasing hormones that then bind at the anterior pituitary, causing it to release stimulating hormones. But we also said that when we talk about this posterior pituitary, it's really just an extension of this hypothalamus. It's the same tissue type. So if you have a cell body up here and an axon that's traveling down, it's just going to travel right through the posterior pituitary and it's going to release into blood vessels. This is vasopressin. There's only two options. There's only two things that get released at the posterior pituitary. One is oxytocin, which we'll talk about in repro, and then the other one is this vasopressin. So vasopressin got released, it gets picked up in the blood, and it gets moved everywhere. So, vasopressin is released when hypothalamus senses high osmolarity. Vasopressin, because it's coming from the pituitary, has to be peptide. We said that everything that comes from the hypothalamus, the anterior pituitary, and the posterior pituitary was a peptide hormone that would bind to GPCRs. Okay. But it's going to go through the blood, and it's eventually going to get down to the collecting duct. So there's our collecting duct there. We're just going to draw it in. Vasopressin went through the whole system, and eventually it ends up getting into the vasa recta. Okay, so that is vasopressin. Vasopressin will move out of the vasa recta into the interstitial space, and then it can bind to GPCRs on the apical side of the collecting duct. So we do the same thing over here. Okay, so vasopressin binds to those GPCRs in the collecting duct. We said there's a lot of different functions a GPCR can have. It's a second message system. We talked a lot about how we can increase contraction. You know, we talked about a lot of GPCRs involved in contraction of smooth muscle, skeletal muscle, all that kind of stuff. But way back when, we had said that it can also do things like increase or decrease endo and exocytosis. And that's what's going to be important here. So within this cell, in this collecting duct, you're going to be making aquaporins all the time. And those aquaporins are transmembrane proteins. So they got made on a membrane-bound ribosome at the ER, got moved to Golgi apparatus, and when they're ready to go, they're actually in the membrane of this vesicle. When you trigger the vasopressin GPCRs, it will cause an increase in exocytosis of those vesicles. So now you merge with the membrane and you have those aquaporins. But you're going to do this on both sides because you have aquaporins in both your apical and basilar membrane. So I really should have done this on both sides. There we go. So now we're just increasing the number of these aquaporin 2s that we have. That gives us more pathways for water to move through the cell and out into the interstitial space to get picked up by the vasorecta. Not only do we have a pathway now, we've got more of a gradient because we put a whole bunch of salt into our blood through the aldosterone story. We increase the osmolarity. So we've got an increased gradient to move the water. It wants to follow. So by pumping that salt in, we created a pathway to pick up more water. And that extra water now is going to be increasing the volume. Okay, so we can increase pressure without having to clamp down on the smooth muscle around our arterioles and arteries all the time. In our story, that's still not going to last very long if no one stopped the bleeding, because eventually he's just going to bleed all this out too, and you're going to run out of water in the blood to reabsorb. That's just it. It's going to be the end. But if you can stop his bleeding, if you can keep him from losing any more blood, and you can pick up as much water as possible to try to keep that volume up, you can probably keep him alive until you can actually do surgery and fix things. But I will remind you that even though that's a nice way to tell the story of how this happens, very rarely do we use it in such an extreme circumstance. Hopefully no one actually ends up stabbed and having to use these systems. But we do use them daily, just as modulation of blood pressure. Okay, so just a little bit up and down to keep things in check. One other thing I really want to point out is it makes it nice to tell all of this in conjunction as one story, but it doesn't have to be. Okay, so the renin can be completely separate from the aldosterone and the vasopressin. Aldosterone, we said here, was being released because renin was binding to the adrenal gland. That's true, I'm not lying, it happens. But it's not the only way we release aldosterone. So there's other things aldosterone could do and other ways to stimulate it. Doesn't always have to be a part of the renin story. Same thing with vasopressin. It could be released by that osmolarity, but it could be released by a whole bunch of other stuff too. And it does other things than what we're telling you. So this doesn't always have to be part of this story. But for this class, it makes it really nice to just tell the story of renin to aldosterone to vasopressin to increase blood pressure. And that's how we'll ask you about it. So that's how you should study through. Questions on? Oh yeah, go ahead. The last thing I'm going to point out is that this system is phenomenally complicated, really, but it's also an amazing target for clinical therapies. Okay, especially blood pressure, because that's what we do so much of modulation here. So you probably heard a couple of terms that you may have seen before, like ACE. ACE1 is going to be the thing that's converting angiotensin 1 to angiotensin 2, and that does all of the smooth muscle constriction, which increases blood pressure. If you have high blood pressure, which for most of us is typically the way we go, low blood pressure is not a normal thing anymore, not with the amount of stress and salt we have in our diets. So in order to affect this, instead of like trying to block angiotensin 2, which you could do, you could like block all of its receptors, except we said this was global. This gets in the blood and it goes everywhere. You'd have to block receptors on every smooth muscle of every artery and arteriole in your body. That's a mess. Not to mention, that's not the only thing that those are good for. So side effects. If instead you could just block ACE so that it doesn't convert as much angiotensin 2 in the first place, that's a better way to go. And ACE inhibitors are actually our first line now for blood pressure treatment. It used to be that they used more diuretics. Where's my vasopressin? Okay, so here's vasopressin coming from the brain. Vasopressin is also sometimes called antidiuretic hormone, or ADH. I don't know about the spelling of that, but so an antidiuretic hormone is causing an increase in aquaporin channels that causes an increase in reabsorption of water. When we do that, when we take back more water, we concentrate our urine. There's less volume. If, on the other hand, we did the opposite, a diuretic, maybe by blocking the aquaporins from getting up here on the channel, we don't reabsorb as much of the water. More of the water now moves through and becomes part of your urine. So if you know anyone who's on a diuretic, you know, especially in the beginning, they pee all the time, because they're blocking the ability to reabsorb as much of that water and instead just flushing it out of the system. By doing that, they decrease their total blood volume, and that reduces blood pressure. Okay. So there's actually a number of ways, new drugs that they're coming out with all the time, targeting different portions of our nephron and collecting duct in order to reduce blood volume and blood pressure in individuals. So questions on the system? Yes?
I don't even know if this is true, but I heard that alcohol makes you pee more, not just because you're drinking, but because it has some effect on you. Is that an effect that you can illustrate within the story you just told us?
As far as I know, alcohol actually acts as a diuretic. Okay, so it's going to be causing more fluid to come out. If it's also having an effect on your brain, it's probably also affecting certain areas that may make you feel like you need to pee more, even if you don't necessarily have an increased volume. I'm pulling that one out of my ears, but yeah.
So you said that vasopressin is an antidiuretic, so it will pull out, or will stop water being pulled into the blood, but won't that also increase the osmolarity in the blood, resulting in more vasopressin being released?
So vasopressin is an antidiuretic hormone.
Yeah.
Diuretic will make you pee more. Antidiuretic will concentrate. So this is going to increase your aquaporin to hold back more water.
Okay.
Whereas a diuretic would be a drug that you could take that would block the aquaporins.
Okay.
Yeah.
Thank you.
Yeah. Anything else? Yes.
If someone does have low blood pressure, would they in general have like a high concentration of angiotensin 2 in their blood, or would it just kind of be like you're at low blood pressure all the time and your body can't compensate for it?
So if you have low blood pressure, and it does happen, it's usually more temporary, transient, especially like when you stand up and you have syncope. But if you did have like a constant low blood pressure, which could be caused by medications, then yes, you would absolutely expect that renin-angiotensin system to be kicking in more, and so you would probably be able to see more angiotensin 2 in their blood. Yeah. Anything else? Okay, we're going to wrap it up by saying we got him to the hospital on time and he survived. Yay! So everybody have a great day. See you on Wednesday.